Okay, let's get started for nucleic acid part 2. Okay, last time we studied the structures of nucleic acids, mainly DNA structures. Okay, what are the building blocks? What are the structures of nucleotides? And how do we classify the nucleotides? How do we name the nucleotides? And then we talk about the DNA double helix structure. And then we talk about the replication of DNA. Okay, what is template-based semi-conservative replication? And also at the replication fork, the leading strand and the lagging strand are actually synthesized differently. Okay, one is continuous synthesized, and other one's synthesized in fragments, and later on joined together by ligands. Okay, today we're going to take a look how genetic information becomes protein. Remember, nucleic acid, the main function of them are to store genetic information. But living things, like human, are in expression of what? Of protein. Okay, our skin, muscle, hair, eye color, everything is expressed in the form of protein. So the information of DNA, the genetic information in DNA, will have to be eventually become what? Form of protein. So that's the transfer of information. Okay, that's the information transfer. And the information transfer is through this very simple chart. Even simple, but again, it actually summarized everything about biochemistry. Okay, this is biochemistry. What is biochemistry? It's basically the transfer of the information from DNA to protein. Okay, that's why we call this transfer the central dogma of molecular biology or biochemistry. How information is transferred to protein. Now, as you can see here, the information is not transferred directly. It is actually through an intermediate molecule called what? RNA. Okay, RNA molecules are heavily involved in the transfer process. Okay, so that means the information is not told to protein synthesis directly, but in the form of what? RNA. So the central dogma is divided into two steps. The first step is the information transfer from DNA to what? To RNA. And then RNA is the guy that directs protein synthesis. Okay, protein synthesis. So today we're going to study these two steps. The first step, transcription. Second step, translation. And after you study these two steps, you will see why they're named transcription and translation. But before we do that, let's take a look at the intermediate molecule, RNA. Okay, what are RNAs? What types of RNAs are involved in this whole process? Okay, first, RNA are similar to DNA. They're polymers of nucleotides. Okay, nucleotides. But they are different in these aspects. Okay, in these aspects. The first difference we studied before, the sugar is different. In DNA, sugar is what? Deoxyribose. In RNA, the sugars are all ribose. Okay, that's the biggest difference. Their sugars are different. The second difference is the base. The T in DNA is replaced with U in RNA. If you guys remember the structure in T and U, thymine and uracil, you find out the structure are basically what? The same. And you also remember in complementary base pairs in DNA, A is paired with what? With T. Now, in RNA, because there's no T, it is replaced with U, so we can anticipate U will also pair with what? A. Pair with A. Because A, U, and T are basically very structurally similar. The methyl group is not involved in hydrogen bonding. So A, if compared with T, A can pair the same way with U when RNA is involved. Okay, number 3, RNA we know are double helix structured. DNA is double helix structured. RNA usually is single strand. Okay, that means there's no second strand for RNA molecules. Okay, there may be some pairs originally from the same strand, but no double helix structure. And because of that, RNA usually are very, very unstable. Okay, they are rarely decomposed, hydrolyzed. And you can imagine that because RNA is what? Is an intermediate. We don't need them to stay long. And we only need them to what? To serve its function and what? And be degraded and be used again to synthesize new ones. So RNA molecules are very difficult to handle in a lab, in a biochemistry lab, RNA molecules, because they're extremely unstable. Okay, why unstable? Because they're single strand. Okay, and the last one, RNA molecules are normally much smaller than DNA. Okay, DNA is a huge molecule. RNA molecules are small, ranging from 75 nucleotides to a few thousand nucleotides. Okay, compared to DNA, RNAs are very small. So these are a few differences between RNA and DNAs. We good? Know exactly everything, like I say, in this chapter. You have to know everything. Next, because RNAs are involved in the process of the genetic information transfer in the central dogma, there are more than one types of RNAs in that chain. And these are the 5 types of RNA we're going to study today. And you will see all 5 of them in the process of genetic information transfer. Okay, those 5 RNAs, their name and their abbreviation, you have to know them all. Okay, but when we use them, we just call their abbreviations a lot more than their full name. Okay, first one, heterogeneous RNA, nuclear RNA, HNRNA. Second one, messenger RNA, mRNA. Small nuclear RNA, snRNA. Ribosomal RNA, rRNA. And finally, transfer RNA, tRNA. Good? Take a brief look at their names. Again, you won't worry about their names because we're going to see them many times in this process. Okay, now after you know these RNAs, you're going to take a look the central dogma again in a different format. Okay, where the transcription is happening and where a translation is happening, and also what types of RNA are involved in transcription versus translation. Okay, take a look at the first. In the first transcription process, it's happening in where? In the nucleus. Okay, inside the nucleus. The information is transferred from DNA to what? To messenger RNA. So what do we get eventually from the transcription step is a messenger RNA molecule. And the RNAs are involved in the transcription steps are these 3 types. HNRNA, snRNA, HN stands for what? Heterogeneous nuclear RNA. snRNA stands for small nuclear RNA. And of course, the final product of transcription is what? Messenger RNA. Okay, these are the 3 involved in the transcription process. The second step, translation process, happens in the cytoplasm, means outside the nucleus. Okay, inside the cytoplasm of the cell. And the 3 RNAs are involved is, of course, messenger RNA, which, again, directs protein synthesis. And then in that step, you need rRNA, ribosomal RNA, and tRNA, transfer RNA. Okay, again, these are this summary showing what happens. Again, don't worry about these details. You don't have to memorize them now. You will see them when we study the detailed steps. Okay, now let's take a look. When DNA is about to get transcribed, normally it's not the whole piece of DNA is get transcribed. When the transcription happens, they're happening at fragments. So a certain piece of information, owning a certain piece, sometimes needs to be transcribed, not the whole piece of DNA. And that certain piece of DNA that is going to be transcribed are that piece that contains information for one protein synthesis. Remember, DNA transferred to what? To a protein. And a whole DNA molecule may can have a lot of protein information, but sometimes you don't have to transcribe them all. You only transcribe a piece of DNA that will be responsible for what? One protein. That piece of DNA has the information for one protein. And that piece is called a gene. Okay, you guys have heard this name a lot, a gene. Okay, G-E-N-E. A gene is a piece of DNA that has the information what? For one protein. Okay, it contains the information for a protein. Now, gene, a piece of gene is also segmented. There's small fragments too, called exons and introns. Okay, this, for example, if this is a piece of gene, that's the information for a protein. The genes is segmented, containing exons and introns. Okay, you can keep those two names in hand. We're going to see them again. Okay, why they're exons, why they're exons and introns. What are exons? Why they're called exon? Why some are called introns? You will understand, but keep that in mind. We can turn back to remind you. Okay, now, after you know what is a gene, let's take a look at something very interesting about the genome. Okay, what is genome? Genome is what? The whole library of genes in a species is called a genome. And if you look at the size of the genome for these species, human, fly, worm, okay, this is one type of worm, C. elegans, and yeast, a tiny single cell, eukaryotic cell, of course. Okay, you can take a look at the genes. Human, we have around 22,000 to 25,000 genes. A fly has 20,000 genes. So what does it tell you? The size of genome for human isn't that much more than what? Than a fly. But apparently we're what? We're a lot more advanced than a fly. So you may wonder, hey, you know, a gene directs a protein. But why we only have maybe 20 or 30 or maybe 3, 4, or 5,000 more genes than a fly, but why our protein is apparently what? A lot more than what? Than a fly. Okay, that's one question we're going to answer after we study the 2 steps. Okay, you will see that why genome size, we aren't that advanced. But eventually when we become proteins, we have way more proteins than a fly or a worm. Okay, you will see that after we study these 2 steps. Okay, so let's take a look at transcription of a gene. Okay, what is transcription? Okay, what happened in a transcription? Okay, then I'll explain to you why we call that transcription. During transcription, a gene, remember DNA is double helix. So during transcription, the double helix has to what? Open up. We call that unwind. Okay, the double helix has to be unwinded by an enzyme called RNA polymerase. After it unwinds, you have 2 strands. Is that right, guys? And you know these 2 strands are anti-parallel, right? One is from 5 to 3, another one is what? From 3 to 5. Okay, we have 2 strands. But let me ask you, how many strands for RNA? One strand. So that means you won't use both, because RNA only has 1 strand. So between these 2 strands, the one that is used to guide RNA synthesis is the one from 3 to 5'. We call this strand template strand. Sometimes you call it antisense strand. Okay, template strand is antisense strand. Because of the template strand is from 3 to 5, so RNA synthesis can be synthesized also anti-parallelly from where? From 3 to 5. Do you guys see that? How do they synthesize it? Through what? Base pairs. G pair with C, A pair with U. Okay, don't forget. Okay, when you write RNA sequence, if you put T here, even though I know you how to do it, it's wrong because RNA doesn't have T. It's just some common mistake. Okay, through base pairs, you can make a chain of RNA. Okay, make a chain of RNA. Now think about it. This strand is called template strand. When you make an RNA of it, this RNA is what? Complementary to this piece. Is that right? These are complementary. And the original, this guy, the blue DNA is also what? Complementary, because they were double helix. So what does it tell you? The sequence of the new RNA will be what? The same as the sequence of what? Of this one from 5 to 3. Does this make sense? Why they're the same? Because they're both complementary to what? To the template strand. Does it make sense? Okay, that is why we call this step transcription. What does transcription mean? Copy. What information is copied? The information in here is copied to where? To the RNA. Okay, they should have the same sequence, except what? T replaced with what? With U. But the sequence of these two should be the same, because they're both made by what? With that template. Okay, this strand is called template strand. This strand is called the informational strand. Why informational? Because it has the information. And finally, the product RNA is called hnRNA, heterogeneous nuclear RNA, which is the initial product of transcription. Does it make sense, guys? Okay, if not, let's take a look real sequence. You may have a better idea. Okay, this is the piece of gene. Okay, like I said, template strand sometimes is also called antisense strand. This is a template. How do we know it is template or antisense? It's from where? 3 to 5. That's the template strand. The other strand, informational strand, sometimes is called sense strand. Okay, they're both used. Okay, actually, sense and antisense is more common in scientific literatures. But your book use template and informational, either one. Okay, you can see that after the unwind, which one is a template? The antisense strand is template, and you make an RNA, A pair with T, U pair with A, C pair with G, and after that, you get hnRNA. And take a look at the sequence of hnRNA. Is that the same as the informational strand? AU-ACCA, AT-ACCA. Again, except what? U replaced with what? T. You can always verify if you made a sequence right. Okay, or sometimes you don't even need to make the pair. Just write the sequence same as what? As the informational strand. Because after transcription, the hnRNA should have the same sequence as what? As the informational strand. Does it make sense, guys? Let me know if it doesn't.
What happens to the informational strand?
They will, after you make this gene, they will go back, like a rewind to make a double helix.
Can you say that one more time?
Say which one?
Like, just, I don't know, I'm just confused. I don't know.
This piece is the antisense strand,
Yeah.
called template. What does template mean? Template means you use this template to make the RNA through what? Base pair.
Right.
How do we know this is template? The template is always from what? 3' to 5'. And the RNA you make is from 5' to 3'. And after you make the RNA, you will find out the sequence of the RNA should be the same as the one that is not used as the template. Remember, DNA has 2 strands. One is as a template, the other one just stay there not to use a template. And the sequence of the RNA should be the same as what? The sequence of the other strand. We call this informational strand. It is not used as a template. Is that the sequence the same? Except what? U replaced with T, but everything else will be the same. Make sense? And the product is called hnRNA. From now, transcription is basically done. After this, the hnRNA will be further modified. We call post-transcriptional modification or processing to give you mature messenger RNA. Does that make sense, guys? We good? Okay, of course, hnRNA here is from 5' to 3'. You should label the ends as well. Always label the ends. Okay? If you're still confused, let's take a look at this practice. See if you can understand. Again, the questions themselves are easy. First is you have to be able to understand the question and catch up the information you need to answer the question. Okay, the following DNA strand is used as a template. They ask you, what is the informational strand? And also write the RNA strand that will be produced. So 2 questions. First, the given strand from 3 to 5 is C, G, T, A, A, G, C, G, G, C, T, G, C, G, G, C, T. 5. Is that right, guys? This is what? Template strand. Is that right? Remember, when you hear this word, template strand is on what? On DNA. Okay, you have to first know, okay, what strand is on what? What is their mention? Which nucleic acid they're talking about? So the first question they ask you, what is the informational strand or sense strand of that sequence, based on that sequence? We know this is the template strand, means what? The other strand of DNA from 5 to 3, then we make it G, C, A, T, T, C, G, C, C, G, A. Did I make it right? Did I make it right? So after I make this is what? The new one I just put down here is what? Did I answer one of the question or not? And make a complementary strand for this given one. The given one is the template. And I make a complementary strand. And this is apparently DNA because T in it, right? So did I answer one of the questions with the new one I wrote? Or not? Not yet. Not yet?
Not yet. You have to replace the
T with the U, right?
Yeah.
There are 2 questions, right? Did I answer one of the questions?
Yes, the first part.
Yes, the first question is answered. Is that right?
Yes.
What is the informational strand? The informational strand is the DNA strand that what? That is complementary to the template strand. So this one is the what? Informational strand. Or you can call it sense strand. Does it make sense, guys? Does it make sense? Good? Okay, again, you have to be very clear. You have to at least know what the question is talking about. A piece of gene has 2 strands. The template strand and what? The information strand. Again, make sure you label the ends. If you label the ends, you will never confuse. Are we good? Okay, now, the second question asks you, write the RNA strand. Okay, there are 2 methods of writing the RNA strand. We know RNA strand is based on what? The template strand. So you can make a paired one. You can make a paired one with that template. Or easier way is to simply what?
Use the information.
Write the same sequence of the informational except replacing T with what? With U. That's easier because you just write from left to right. You don't have to make pairs. So the RNA will look like, again, 5, G, C, A, U, U, C, G, C, C, G, A, 3'. This is what? The HNRNA string. Should be the same sequence as what? As the informational string. Except, again, don't forget replacing T with what? With U, guys. Very important. This is a very common mistake. Sometimes you just ignore it and just read the same sequence. RNA doesn't have T. Okay, this is principle. I know you know it, but principle wrong. Question-wise is very simple, right, guys? And then you have to be very careful when dealing with questions of this chapter. Please let me know if it makes sense. Guys, all of you, please let me know if it makes sense. And also you can verify that the hnRNA strand will be complementary to what? To the template. You can see that G with C, G with C, A with T, A with U, G with C should be complementary. But like I said, if you know the informational strand, the easiest way is just write down the informational strand, replacing T with what? With U. Good? Okay, you have to know their names. Okay, these names, because sometimes the solution is easy. The things you have to know where that is. Template is on DNA. Informational on what? DNA. RNA has U, no T. And the sequence of RNA should be the same as, again, informational strand. Okay, I will use more information than template than sense and antisense in your quiz and tests, because that's how what your book is used. Does it make sense, guys?
So you'll use informational on the
And template. These two names instead of sense and antisense. Okay, more, because I know your book uses these two. Now, like I said, after this transcription step, this is transcription. Again, what is transcription? Because you get a sequence the same as what? Information. That's why we transcribe. You basically what? Made a copy. And after this, like I said, the following steps are called post-transcriptional. Post means what? The transcription basically done. You just make more modification. And that modification is very essential and partly answer the question why we're more advanced than a fly. Okay, let's take a look. What is going on during post-transcriptional modification? Remember we said the piece of gene is segmented, right? Has what? Exon and introns. So when it's transcribed, remember transcribe is what? Make an exact copy. So the hnRNA also should have what? Exon intron, because they remember the sequences are the same. If gene has exon intron, hnRNA has what? Exon intron. During this step, okay, during post-transcriptional modification, the introns will be cut off, taken away, and the exons will be joined together. After the exons join together, you get a mature RNA product. We call that messenger RNA. And the removing introns, joining exons is cut, tater. This is like a little tater, later workers, guys doing this job. It's done by SN RNA, small nuclear RNA. They're basically RNA enzymes. Okay, this is a very important step. Like edit the information of hnRNA into what? Into messenger RNA. So when you see later practice, we're going to do that next Monday. Okay, when you have intron exons, after you do the transcription, you need to what? Remove the introns and only keep what? The exons joined together. Why do we do that? Why nature designed this way of putting some spacers between the information? Why do we do that? Take a look next picture, you will understand. Okay, this is a picture showing a piece of hnRNA with exons and introns. You can see there are 4 pieces of exons and 3 introns. Okay, for example, when the introns are removed, when joining exons, we have 4 pieces of exons, but when we join them, take a look. We have what? many ways of doing that. Is that right? First way, we only join what? These 3 exons. The second one, we join these 3 exons. This one we join all together. This one we only take these 2. So what does it tell you? You have a complete sentence. You insert some useless words. But when you give that information to someone, they have a different way of what? Interpret it. So this gives us what? A lot more messenger RNA that you can imagine from one single piece of hnRNA. So the library actually what? Expanded, right? This is a gene, but the gene can actually result what? Many proteins. Remember, who directs protein synthesis? Messenger RNA. So now from one hnRNA, you can have what? Many messenger RNA for what? For many protein. So even our genome is not as many as, not really more than a fly, but because of this, our messenger RNA library what? Expands a lot. Okay, messenger RNA expands a lot means what? Our protein expands a lot. So you can see that genome is around 22,000. But because of that post-translational modification, our transcriptome, the messenger RNA library is what? Increase a lot. Okay, increase a lot. Later on, after that, we're going to take a look. Another further jump over. Our proteins are way more than a fly because of this. And this step is called alternative splicing. Okay, what is alternative splicing? It means what? You have different ways of splicing things. Like you're making a clothes, right? You cut the one you don't need and you join the one you need, you can make a different clothes. Okay, this is transcription. Does it make sense, guys? Okay, next, let's take a look. Translation. Before we know translation, we first need to take a look at the mature product, messenger RNA. Okay, messenger RNA. By the way, you guys, how many of you have COVID vaccine? All of you guys, or most of you guys? That's why I'm not judging or anything. I just want to tell you, COVID vaccine, you guys know is what vaccine? Messenger RNA vaccine. You're basically getting the messenger RNA from the COVID virus. And your body will synthesize a piece of COVID virus protein so that you can get the antibodies of the protein, of the virus. Okay, that's how it works. Okay, this is I think the first case messenger RNA vaccines are used. Okay, other countries don't use it, but Pfizer and Moderna, they are the same. They're both messenger RNA vaccines. Okay, now what is messenger RNA? Messenger RNA is the one that directs protein synthesis. Okay, now, if you look at the messenger RNA, we know the information messenger RNA has is from what? From DNA, because it's transcripted after the transcription. And the information, okay, the information is in what? In the sequence of what? The base. Is that right? We said the genetic information. What is genetic information? Sequence of what? ACGT, the base sequence. So in the piece of messenger RNA, it actually has some genetic codes. Okay, genetic codes. They're in a group of 3 bases. So from 5' to 3' of RNA, you will have a lot of codes in a group of 3 bases, or group of 3 nucleotides, of course. And these codes are called genetic codons. They're in what? In messenger RNA. And each codon contains what? 3 bases. Again, reading from 5 to 3. Okay, the direction is also important. You will see that in your practice and exercise. What is the codon for? The codon will tell you, hey, If I have a codon here, then what amino acid do you need? Remember, proteins are made by what? Amino acid, right? So the codon here actually tells people which amino acid is first, which amino acid is second, which amino acid is third, so you can make a what? You can make a protein. And so far, we have deciphered that codon. Okay, it's not a secret. We know the genetic codon very well. And this is the chart of the codon. Okay, you will be provided this chart during your test and quizzes. Okay, you don't have to memorize the codon, but you have to know how to use the chart. Remember, the codon is what? A group of 3 bases reading from what? 5 to 3. So if you know a codon 3 bases, all you need is find which one is the first, which one is the second, which one is the third. Then they will tell you, hey, for a codon, for example, ACU, ACU. Then this codon ACU what? Tells you, oh, this codon means you need a threonine, which is what? An amino acid. Or AGU, for example, AGU means what? Serine. Easy? This chart is very easy to use. Okay, you do notify that, hey, why all these codons tell you the same amino acid? Because codons are degenerated. Why is that? Think about, for a 3-base codon, how many possibilities are we? Or how many codons do you think we have? A codon has 3 bases. Each base has how many options? Each base, 4, right? ACGT. So 4 times 4 times 4. There are 4 times 4 times 4. 64 codons. We do have 64 codons. But tell me, how many amino acids do we have?
56?
Amino acids?
20 amino acids.
20 amino acids, right? We have more codons than what? The amino acid. That's why a few codons will tell what? The same amino acids. It's a simple reason. Okay, here are some facts about the codons. Okay, first, the codon is highly degenerate. That is, many amino acids are designated by semicolon. Okay, we connect synonyms. The same reason. Okay, because they have more codons than amino acid. Okay, all synonyms of an amino acid fall within a single box, and the first 2 bases will be the same. Okay, what does it mean? It means, for example, these are synonyms for what? Arginine. You can see the first 2 letters are what? The same. Okay, this is very common for degenerated codons. Okay, out of this 64 codons, 3 codons don't tell an amino acid. They are either initial codon or stop codon. We're going to see that later on. Okay, see that these 2 are stop codons. That's a stop codon. Okay, this one is initial codon, but initial codon normally designs an amino acid. So 3 codons actually don't designate an amino acid. They're stop codons. Stop codon means later on you will see that tell the factory to, hey, your protein's done. Stop here. And finally, okay, why I want you guys to use that chart, because the genetic codon is universal. Means what? We use that codon. Most species on this planet use that codon as well. Okay, that's a universal codon chart. Good? Right, after we know the codes on messenger RNA, let's take a look. What is translation? Okay, translation means what? I'm making protein now. But remember, the codons are in the sequence of what? Bases. But amino acids are in the sequence of what? I'm sorry, proteins are in the sequence of what? Amino acid. So you're looking at 2 sequences, 2 languages. Okay, 2 languages. So that means in order to read the codon and make the protein, you need somebody what? To know both language. One language is the codon, another language is what? Is the sequence of making the sequence of amino acid. And that translator, the guy who interpreter, the guy who knows both languages, is another RNA molecule called transfer RNA. Okay, from this picture you can already see that. Okay, transfer RNA, this is a transfer RNA. One end of it will what? Read the codon. The other end will what? Bring an amino acid. This is amino acid, and here's the protein growing. Do you see that, guys? Okay, this is another transfer RNA is going to read the codon, and then what? Bring another amino acid. That's why they get their names. Transfer RNA. Transfer what? Amino acid. Let's take a look. Okay, this is a picture of a transfer RNA molecule. They're very small. Transfer RNA is the smallest. And one end of it, it's like a hairpin structure. Okay, you can see that it's still single strand, but locally there are some base pairs. Okay, some loop, some base pairs, but a single strand. Okay, and one end of it, it has a 3 base group as well. And this 3 base group is going to read the codon. What does it mean, read the codon? Read the codon means the 3 group is going to pair with the what? With the codon. So here you can see that CUU, for example, this is the parts they read, that their ears or mouth or whatever. Then the codon here will be what? GAA. We call these 3 bases on transfer RNA the anti-codon. Remember, codon is on what? Messenger RNA. Anti-codon is on what? tRNA. And on the other end of it, the 3' end, it will carry an amino acid. Which amino acid it carries is determined by what, guys? Let me ask you, see if you catch all the information. Who decides the amino acid, the identity of the amino acid here? Who decides? We just studied a few minutes ago. What decides the identity of an amino acid? The what? The codon, right? Where is the codon?
The template.
The messenger RNA, right? So, the codon here, it doesn't show. CUU pair with what? Again, GAA. The GAA here tells you, hey, what amino acid do we need to bring? Because the CUU is going to what? Pair with it. Okay, don't get confused. A lot of times you get confused. Not this one. This one is read the codon. The codon tells people which amino acid. Let's go back. We can go back to check. GAA, which amino acid? And you guys take a look very quick. You can use the chart. GAA, if I tell you GAA, which codon? Which amino acid? Glutamate what? Acid. So that means this transfer RNA, this particular guy, which has the anti-codon to read the GAA, will have to bring what? Glutamic acid. Nothing else. He will only bring that. So don't get confused again. Not this guy determines. That's anti-codon, fine. But that reads the codon. It's the codon on messenger RNA directs what? Directs amino acid. Okay, directs amino acid. Okay, with amino acid, when amino acid is bonded with amino acid, bonded with the 3' end of the RNA, transfer RNA, they're actually linked through an ester bond. Okay, they're linked through an ester bond. Remember, 3' has an oxygen on there. Okay, the oxygen of the sugar, it bonds through an ester bond with the amino acid. That's why they can bring an amino acid. Okay, they're bonded, covalently bonded. Okay, again, codon, anti-codon. It's the codon here directs what? Amino acid. The codon is on what? Messenger RNA from 5 to 3. When you read the codon, you must make sure you need what? 5 to 3. That's very important. Okay, because UCA, ACU are two different codons. Okay, make sure you read from what? 5 to 3. Okay, not the anti-codon. It's the codon directs the amino acid. We good? Okay, now after that, we'll watch a very small video about translation. Okay, after we know all the background of translation, the translation scheme. Okay, I want to show this picture again, and then we watch the video. Looks like this. Okay, this is what basically what translation looks like. This is what? Messenger RNA. Is that right? Has what? A lot of codons. And on the messenger RNA, there's a factory built on it. The factory is not big. The factory is relatively small compared to the size of the messenger RNA. The factory is called the ribosome. Okay, the factory is called a ribosome. A ribosome has two units, a large unit and a small unit. Again, we'll see more, but I want you to point out something. So you watch the video, you can see understand better. Inside the factory, there are two spots. So the factory basically covers two codons, only two codons. Okay, two sites. So that means what? Inside the factory, there are only two what? Amino acid, two transfer RNA reading the codon. And because the factory only covers two codons, if you want to make a protein growing, the factory has to what? Has to move forward. Because each time you can only read what? Two. Like reading a book, you can only read two letters, you have to move what? Forward. But each time it only covers what? Two sites.
So the
Right?
tRNAs are inside the ribosome.
Correct. Inside the ribosome. Two tRNAs. Okay, two tRNAs. Okay, so after you know this background again, the messenger RNA, the factory is called the ribosome, and the factory will cover two codons. Okay, so you have A sites and P sites, we'll see that later. But every time you have two, they will make a peptide bond, and the factories will keep moving forward as your what? As your codons are read, and also the chain growing. So these are some backgrounds, so let's take a look at the video. How translation is done. See that? Transfer RNA comes in, bring amino acid, and you can see this is the protein chain. Is what? It's growing. And after the factory is moving forward, like reading the codon. This is basic translation done. The protein chain is done. The protein chain will be, this time will be get out of the cell. Okay, will be get out of the cell. All right, we can watch this again. Let's take a look at the detailed steps. Okay, like I said, the factory where the proteins is synthesized is called the ribosome. It is a cell organelle, ribosome. Ribosome is a large molecule. Main component of it is RNA. Okay, the major component of it is RNA. 65% by mass is RNA. 35% by mass protein. And the RNA inside the ribosome is named after the organelle. Ribosomal RNA, rRNA. So now we have seen all 5 RNAs, if you can remember. hnRNA, snRNA, messenger RNA, transfer RNA, and what? rRNA. Where's rRNA? In the ribosome. 65% of ribosome is rRNAs. And in fact, the active site of this whole factory is located in a large unit. Okay, in a large unit. And messenger RNA binds to the small unit. And here is the binding of the amino acid with the 3' end of the transfer RNA. You can see that ester linkage. You see that this is amino acid, right? You guys remember structure, amino acid, alpha carbon, base, amino group, acid group, R group, H. Is that right? The acid makes an ester with the 3' end. That's why the amino acid can be carried by transfer RNA. Okay, it's ready to go. So let's take a look. First, like the video shows, the small unit binds to the messenger RNA. Of course, 2 sites, only two codons. And after that, the first transfer RNA comes in. The first codon is usually AUG. It's the initial codon. The first amino acid for eukaryotic cell is methionine. For us, that's normally our first amino acid. Okay, that's why the first codon, initial codon, tells you, hey, this is the first amino acid you need. After the first transfer RNA, again, this is the codon, that's what? Anticodon, right? The codon AUG directs methionine. And after that, large unit comes in, make the factory. Okay, the complex is ready. Then, take a look. The factory contains 2 codons. We call those 2 codons 2 sites. The site on the left is called the P site. The site on the right is called the A site. Okay, I'll explain to you why P and A. You don't have to memorize it. I'll explain to you why P and A. Okay, P site and what? A site. And if remember, the factory is going to what? To move. So whenever we move 2, the left one is always the P site, the right one is always what? The A site. Okay, you keep that in mind. After the factory is ready, the second transfer RNA will what? Will come in. Will read the codon. Of course, the second codon here is GGU. GGU tells glycine. So you can see that we have 2 amino acids ready, right? This is amino acid ready on 2 codons. Okay, after that, they will make a peptide bond. Okay, they will make a peptide bond. What happened is the amino acid on the P site will be transferred onto the amino acid on the A site to make the peptide bond. Here. Okay, P site, A site. This is the ester. The acid from the ester will transfer onto the nitrogen to make what? To make the peptide bond. CON. You guys see that? Okay, you don't need, I won't ask you for the reaction here. I just want to know what's going on, what happened. Okay, the transfer is always from P site to A site. Whatever on the P site will be transferred onto what? Onto the A site to make the peptide bond. And after that, take a look. We now have a what? Dipeptide on the A site, right? And then the P site is what? Empty, because the amino acid is gone. Do you guys see that? After that, the factory is going to what? Shift one spot, cover this site and a new site. After it shifts, okay, after it shifts, this is what happens. This guy will be gone, and the factory will shift. A new transfer RNA will come in. Okay, new transfer RNA coming. your factory will look like this now. Does it make sense, guys? See that? The A site has how many amino acids? 2 now. And after it shifts, like I said, as long as the site on the left is called what? P site. So now the original A site becomes what? P site. And the new one becomes what? The A again. Now take a look. Now you will understand why these names P and A. On this site, you have a what? Peptide. Is that right? You always have a peptide. On the A site is always what? The new amino acid. That's why this is called the P site. P stands for what? Peptide. A stands for what? Amino acid. Think about when you shift a new copy, the new one is always a single what? Amino acid. The chain is always on the P site. Does it make sense? And of course, what this happens, the transfer is again from what to what? From P site to what? To A site again. Now you have a free peptide. Does it make sense? After this guy transferred, always from a peptide transfer onto a single guy to make the chain longer. Okay, to make the chain longer. So after that, you can see that the transfer happened again, and the fourth guy will what? Come in, and so on and so on and so on. Does it make sense? Okay, this is a very interesting, amazing step. Okay, and again, you always have 2 sides. The left side is always have a longer chain. The right side is always what? A single amino acid. That's why P and A. P stands for peptide. And the transfer direction is always, no matter how long this is always from where? From the peptide to a single amino acid. Like you're making a necklace, you always use the chain of necklace to add another bead, not adding bead to the chain. And finally, when the codon is one of those 3, UAA, UAG, UGA, which is a stop codon, and at that point, a stop factor will bind to cleave the protein off the factor. Okay, this is translation. Okay, now you understand what is translation. Translation is basically change the language of the base codons to what? To the sequence of amino acid, to languages. That's why we call that translation. Okay, here is the picture showing you what we've just talked about. P site, A site, transfer from P to A, you get what? Dipeptide. Then shift, you got something like this. The A site here becomes the new P site. The newly one is the new A site. Transfer the dipeptide to the tripeptide again, and new guys coming again. So you just keep reading and keep elongating the chain. Okay. And of course, to increase, yes?
Sorry. You said the translation was basically to change language of base codon to what?
To the sequence of amino acids.
Okay.
Because protein is made by what? By amino acids.
Okay. Thank you.
Yeah. That's why we call translation, because they're 2 different languages. Okay, codons is in the sequence of what? Bases, ACGT. Protein is in the sequence of what? Amino acid orders. Okay. And of course, to increase the efficiency, multiple ribosomes are actually binding to the messenger RNA. You don't just have one, you have multiple ones. Okay, like people are keep reading it, making more proteins. So at the same time, you have many proteins. Okay, not just one. You can see something would look like this. Okay. A lot of people are reading the same one to make proteins at the same time, right? So I'll let you guys to watch this again. Okay, see how the protein is. Now you have a better idea what is going. You can see the peptide is always transferred onto the new one. Grow. Finally, after the protein is made from translation, okay, from translation, we have a lot of things can be done on that protein called post-translational modification. We can put a phosphate on it, we can put metal on it, we can put a sugar on it, we can put other protein or hydration or method. We can put a lot of things on the protein. So we go back to this chart again. Okay, even though we can make this many proteins, but we can do modification on that. Take a look. Our genome is 22,000 genes. When it becomes messenger RNA, the number becomes 200,000. When it comes to proteins, the number is over 1 million. That explained, even though our genes aren't a lot more than a fly, but our proteins are more what? Diversified. Okay, functional diversified. That explained why. Alright? That is all for this chapter. I don't want to do the practice today. The reason is I want you to guys digest the content. And especially next Monday, after a few days, I want you to see you guys still remember or catch up anything we talk about this class in a real practice. I know understanding that these things are simple, but when it comes to ask you a question based on the content, it's a whole different story, trust me. Okay, so next Monday, we're gonna practice 2 major questions, and you can bring your questions as well. And I want to see you guys how you work on these questions. Okay, so let me stop the